My name is Steve Laviolette, and I wanted to share with you today some of the research our lab's been doing looking at the effects of adolescent nicotine exposure on long-term disruption of mesocortical limbic function and their relationship to increased risk for anxiety and mood-related disturbances. Now, we're seeing a very strong increase in the rates of vaping behaviors, especially among Canadian teens recently, particularly during the pandemic lockdowns. We've seen substantial increases in levels of nicotine exposure and dependence, and more concerning the relatively higher levels of nicotine content in a lot of these tobacco-based and vaping products raises questions about how exposure to nicotine during vulnerable adolescent brain development periods might increase risk for certain neuropsychiatric disorders over the long term. So there's a lot of both clinical and preclinical evidence that suggests that nicotine, which is the primary psychoactive compound found in tobacco, strongly interacts with the mesocortical limbic system. And by that I'm referring primarily to the prefrontal cortex, the nucleus accumbens, and ventral tegmental area. And of course, the major neurotransmitter system importantly involved in regulating anxiety and mood disturbances within this brain circuitry is the dopamine neurotransmitter pathway. Now, over 95% of lifetime dependent smokers will begin their habit during their adolescent years. And this is really important because during adolescent brain development, there's a lot of really important synaptic tuning going on, synaptic pruning, connections between the cortical regions and subcortical regions are also taking place, which is important for the ultimate regulation of subcortical emotional processing centers, such as the mesolimbic dopamine pathway. So any sort of external insult via exposure to certain drugs, such as nicotine, during these vulnerable periods of brain development can have long-lasting impacts on psychiatric risk later in life. Now, there's a lot of clinical evidence suggesting that there is a causal link between exposure to nicotine during adolescent brain development and an increased risk of developing mood and anxiety disorders later in life. And just a couple examples of some of the clinical studies that have shown this very strong association between cigarette smoking and the increased risk of developing anxiety disorders and mood disorders later on in life, particularly during young adulthood. Now, using preclinical rodent models of brain development and drug exposure gives us the advantage of being able to control the precise amount of nicotine exposure that's taking place and also to temporarily control the exposure period. So we use a model of rodent brain development wherein we compare both adolescents and adult controls and give them doses of nicotine 3 times daily for 10 days during postnatal day 35 to 45. And this allows us to mimic a lot of the neurodevelopmental processes that are happening in the mammalian brain during this specific window of brain development, and provides an excellent preclinical model of exploring how exposure to nicotine during these vulnerable brain periods can impact later changes to mesocortical limbic function and various behavioral analogs of mood and anxiety type symptoms. Our lab uses a variety of classic preclinical behavioral models in rodents to examine the potential side effects of adolescent nicotine exposure on anxiety and mood type disorders. One of the assays is called the light-dark box. So this basically taps into the animal's preference for a dark environment and the amount of time the animal is willing to spend in the white side, the light side of the compartment, is taken as a measurement of their anxiety behavior. So when we compare adolescent nicotine exposed versus vehicle controls, we see that in the black bars, adolescent nicotine exposure causes significantly less time in the light box. So the animals are experiencing far higher levels of anxiety and increased time in the dark box as well in panel F. The number of transitions in panel G, shown in the black versus gray bars, again, is significantly down in nicotine treatment, suggesting that there is a very strong anxiety type phenotype in these animals when they reach young adulthood. We also looked at subthreshold fear memory. So these are fear conditioning procedures, exposure to a foot shock and a conditioning cue. And what we see in the black bars is that when you're exposed to a subthreshold fear memory that normally is not strong enough to evoke a fear memory, exposure to adolescent nicotine actually potentiates that fear memory formation, suggesting that adolescent nicotine exposure makes you far more sensitive to negative stimuli in your environment, which is a classic endophenotype associated both with anxiety disorders and mood disorders. Now, some additional preclinical behavioral assays for looking at mood type disorders and symptoms using rodents are the forced swim test, the sucrose preference test, and the sociability or social memory test. So what we found was that, again, looking at the black bars, the amount of swimming and immobility time measured in the forced swim test was essentially exposes the rat to a cylinder filled with liquid, and the animal attempts to escape from that environment. The swimming time in the nicotine-exposed animals is significantly reduced, and the immobility time is significantly increased, suggestive of a depressive-like phenotype following adolescent nicotine exposure. Same with sucrose preference. Now, normally a rat will naturally prefer sucrose as a sweet type of substance. It's highly rewarding, but in the nicotine-exposed animals, again, we saw that there was a significant decrease in sucrose preference, and no such effects were observed in the adult exposure, highlighting again the importance of adolescence versus adulthood exposure for a lot of these effects to take place. In terms of sociability, adolescent nicotine-exposed rats spend less time, shown in the D panel there, exposed when confronted with a stranger animal. A normally healthy animal will spend more time with a novel conspecific, but following adolescent nicotine exposure, they spend less time. They have less social motivation. And in terms of social memory, their ability to recall a previous social experience, adolescent nicotine-exposed animals have significant deficits in their social cognition, again, which are very similar to the sorts of psychiatric symptoms we see in people suffering from mood and anxiety disorders. What's really striking is when we went in and did electrophysiological recordings in both the ventral tegmental area, which houses the ventral tegmental area dopamine neurons, and the prefrontal cortex, which has pyramidal neurons and interneurons, which are very important for executive cognitive function, we found that adolescent nicotine exposure caused a hyperactivity response in the VTA dopamine. So in panels B, C, and D, we see a significant increase in the spontaneous firing rates of the ventral tegmental area dopamine neurons, suggesting a hyperactive dopamine neuronal state, which may be some sort of compensatory mechanism associated with anxiety and mood-related disturbances. In the prefrontal cortex, we saw a similar hyperactivity of the firing frequency in the pyramidal neurons in the prefrontal cortex. And this suggests that there's a loss of normal inhibition, perhaps a loss of GABA inhibition in the prefrontal cortex. Again, these sorts of symptoms are characteristic of what we see in mood and anxiety disorders. Now, we looked at a lot of molecular biomarkers in the prefrontal cortical areas of these nicotine versus vehicle-exposed animals. And what we found was that in the prefrontal cortex, taking tissue samples from the prefrontal cortex, nicotine exposure during adolescence led to a significant loss of specifically the dopamine D1 receptor subtype. No changes were observed in D2, but there was a significant loss in D1, and also abnormal levels, hyperactive normal levels of the phosphorylation state of ERK1 and ERK2. Now, both D1 receptors and ERK1 and ERK2 levels are known biomarkers of depression in human clinical studies. So this was an interesting correlate of some of the molecular biomarkers known to be linked to depression and mood disorders that we were able to observe following adolescent nicotine exposure in the rat brain. This is an interesting postmortem study from Derek Cannon showing that there is in fact a loss of the dopamine D1 receptor in several brain areas associated to anxiety and mood disorders, such as the caudate nucleus, the ventral pallidum. We see that loss specifically of the dopamine D1 receptor, and similar to our results, they found no changes in the D2 receptor. So there does appear to be an interesting correlate between loss of D1 receptors in human patients with mood and anxiety disorders to what we're seeing following adolescent nicotine exposure using our animal models. Now, there's significant evidence not only related to the prefrontal cortex and the ventral tegmental area dopamine system, but also to the ventral striatum, which basically corresponds to the nucleus accumbens in both the rodent and the human brain. And this has shown that there is abnormalities in reward processing activities, specifically indicated by dysregulated activity and abnormal metabolic activity in the ventral striatum, and in the reward circuitry associated with the mesolimbic dopamine pathway. So we began a series of studies looking specifically at the ventral striatum and how adolescent nicotine exposure might induce some of these depression and anxiety-related phenotypes, specifically in the ventral striatal region. So in this study, we looked at how adolescent nicotine might induce depressive and anxiogenic type symptoms through regulation of the ERK1/2, AKT, GSK3 pathways, both of which are linked to dopamine transmission and neuronal dysregulation patterns in the nucleus accumbens, again using our rodent model of adolescent nicotine exposure. Now, interestingly, similar to what we saw in the prefrontal cortex, we saw in the nucleus accumbens this hyperactivation of the ERK phosphorylation state, specifically in the nucleus accumbens. We also saw abnormal elevations of the AKT and the GSK3 pathways. Now, these are two molecular biomarkers that are linked both to anxiety disorders and mood disorders, both in terms of their genetic susceptibility and in terms of abnormal regulation of dopamine transmission, GABA, glutamate as well. We also saw significant reductions again in the dopamine D1 receptor and no significant effects in D2. So there's an interesting pattern emerging in terms of abnormalities in the dopamine D1 receptors following adolescent nicotine exposure. Now, we wanted to see how adolescent nicotine exposure might alter neuronal activity patterns within the ventral striatum following exposure during adolescence. So we went in and actually recorded neuronal activity states from the medium spiny neurons, which comprise the majority of neurons in the ventral striatum. And what we found was that adolescent nicotine caused this hyperactivation state of medium spiny neurons directly in the nucleus accumbens. We did some pharmacological analyses, and we found that both the hyper firing frequency and the hyper bursting rates that we found in the medium spiny neurons, both of which indicate dysregulation of neuronal activity in the ventral striatum following adolescent nicotine, they were able to be reversed with a GSK3 inhibitor. So that was the drug in the blue bars there, and we were able to restore these abnormal neuronal phenotypes simply by inhibiting the GSK3 pathway, suggesting that there's a functional correlation between the GSK3 signaling pathway and its ability to dysregulate these neuronal activity states directly in the nucleus accumbens following adolescent nicotine. We've also started some really exciting imaging studies using Maldi Imaging, which is matrix-assisted laser deionization, which basically allows us to image any neurotransmitter or specific metabolite of those neurotransmitters in fixed brain tissue specimens. So in here, these examples were showing Maldi scans of the prefrontal cortex looking at GABA and glutamate. So on the top bar there, we see that there's a significant loss of GABA in the prefrontal cortex following adolescent nicotine exposure, suggestive of the hyperactivity state that we observed previously in the previous slide. Interestingly, we also see a loss of glutamate in the prefrontal cortex using the Maldi imaging specifically following adolescent nicotine exposure, again suggesting that there's this imbalance between GABA and glutamate in the prefrontal cortex, which may be an important biomarker for the comorbidity between adolescent nicotine exposure and increased likelihood of anxiety and depressive-like symptoms. We also saw a significant loss of GABA in the ventral striatum following adolescent nicotine. And again, this is consistent with the hyperactivity of the neuronal firing and bursting rates that we saw in neurons in the nucleus accumbens following adolescent nicotine exposure. And again, this is suggesting that adolescent nicotine exposure specifically leads to alterations in GABA and glutamate imbalance within these regions, which might be an important biomarker for the increased risk of mood and anxiety disorders following adolescent nicotine exposure. So I'll just conclude by talking about some really exciting translational clinical studies that we're now working on based on these rodent preclinical studies that we've conducted and I've talked about previously. And these are studies that we're performing with Dr. Beth Osich, Dr. Jean Theberge, Dr. Matt Park, Dr. Walter Rushlow, and Dr. Rob Hegley. And essentially, we're going to be performing clinical imaging studies in mood and anxiety disorder patients that are recruited from FEMAP, of which Dr. Beth Osich is the director. And we're going to be recruiting patients that have a history of adolescent tobacco dependence. So our studies are going to include magnetic resonance imaging and fMRI of both the prefrontal cortex and the striatum, and also use diffusion tensor imaging to examine potential disruptions in some of the white matter connectivity patterns between these two brain areas and how they might be associated and correlated with mood and anxiety type symptoms following adolescent nicotine exposure. We're going to be looking at changes in glutamate and GABA levels using MRS as biomarkers for these nicotine and mood anxiety disorder comorbidities, again, based on the preliminary preclinical evidence that we've collected from our rodent models. We'll be taking saliva samples, and these will be used to look at genomic markers of nicotine dependence and comorbid mood and anxiety disorder symptoms, and again, largely correlating these back to a lot of the molecular biomarkers that we've been able to identify in our preclinical work. So ultimately, our long-term goals are how can we target these molecular adaptations and these long-term neuropsychiatric side effects, and identifying who might be at greater risk of developing mood and anxiety disorders if they expose themselves to nicotine during these very vulnerable periods of adolescent brain development. So thank you for your time, and I'd just like to acknowledge our funding partners, CIHR, NSERC, MITAX, Schulich, and the Lawson Health Research Institute, and of course the Department of Psychiatry for the seed funding grant that will be used to conduct our initial clinical studies using our imaging work with patients recruited from the FEMAP. So thank you again for your attention.